Now we're going to look at a new way of classifying called, well, by many names, but essentially genetic classification. It goes under the names of cladistics, phylogenetics. Essentially, it's how do we organise and compare organisms and classify them based on the genes and the gene recipes that they have inside them. So this type of classification has essentially rewritten some elements of the book of life, because it's found that many organisms that we thought were quite different because of the way they looked, their morphology, when you actually look at their genetics, they're actually quite similar. So we've actually rearranged where they sit in the tree of life to make sure they're closer to organisms that have similar genetics to them and further away from organisms that have quite different genetics than them. Now, there's a few things when it comes to looking at cladistics. There's a few assumptions that they make that we need to take into account because they're not always correct. Number one is that genetic changes, which we call mutations, have resulted in Now there's a few assumptions that we just have to mention here because those assumptions underpin how we interpret the cladograms and stuff that you're going to be actually having a look at. Assumption number one is that genetic mutation has given rise to all of the different types of organisms that we see today. And in order for that to be true, you have to assume a few things. Number one is that mutations, which are changes to genes over time, resulted in new genetic recipes being created, as well as recipes being changed or destroyed. And it's the new part that is the big contentious issue there, that new genetic code, which is simply code, it's not the actual protein that does the job, it's just the recipe book, that a new recipe can accidentally be created through mutation. So we'll talk a little bit more about that later. The second thing it assumes is that mutations occur at a constant rate. Now this isn't always shown in the fossil records, so they can't quite explain how we've had explosions of species appear all of a sudden. How do we actually compare species at a genetic level? We use the DNA or their protein sequences with a variety of techniques, and we analyse the percent similarity between different species. Now, why do we do this? Because essentially genetics is composed of two things. You've got a blueprint or a recipe book, which on its own is actually useless unless you actually do something with it, because it's simply the written code. That's your DNA. And then you have the actual object that's been made from that recipe book. So if you're cooking, it'd be a cake. The cake is actually what is the functional part. That's what's actually been made from the recipe book. And in organisms, that's protein. So we look at either the recipe book or what it makes. We look at the blueprint or the protein. So we look at DNA sequences and protein sequences and compare them between species, because there's a huge amount of overlap and there's a huge amount of protein that is similar between species, and we look at how different or how similar they are. Now, to investigate how scientists actually look at these similarities and differences, we're going to use two different types of diagrams: a character map or table and a phylogenetic tree diagram. Now, you've most probably seen this one before, but you haven't seen the table one before. Essentially, the table is what gives rise to the diagram. So we're going to be looking at how they actually organise information into this table. Essentially, they're identifying and organising trends, and then we use that to develop the phylogenetic tree diagram. Now, a really good example of this is actually on Bozeman Science. It's a video called Cladogram. So I'm going to get you to stop the video now and go and watch that video and take some notes. And then we're going to come back to this and see not only how he puts it into the phylogenetic tree, but also how he actually, or how that information can first be organised into a character map and table. So we're going to stop this video here, and you're going to go and watch cladograms with Bozeman Science, and then come back and we'll finish the lesson.